Welcome back. I trust you had a productive and effortless lunch. It's been certainly a wonderful day. Some of you got a chance to go outside and explore the day and the biological implications of a beautiful spring day here in Cambridge and at MIT and at the lab. Our final session today will extend on our explorations that began with complicated biologically based but essentially socially networked systems that have as an inspiration dynamics and processes that we find in biology, and trying to make the translation to predicting how those systems work in actual practice. Sometimes biology is a useful metaphor, sometimes it's not, but as a design principle, exploring those complicated systems with nature in mind is a very, very important principle to advancing the research and the science. Then we moved on and opened a window into the brain itself and explored how processes that are inspired from nature, and in fact using processes that neurons already exhibit in various mysterious functions as an invitation to connect with brains and to learn more about how they work. We finish up now with a discussion of life itself, a sense that the biologically based systems upon which all life depend are replicatable, are contained within them design principles and important notions of how we could construct, whether we're talking about synthetic organisms, but certainly creating systems of a sufficient level of complexity that they enable the kinds of things that we think of as life, as natural processes. And so that will be our exploration here in this final segment. A couple bits of housekeeping business that I'll get to in a moment, but we'll start you off as people file in from lunch and get seated by, first of all, once again welcoming Joey Ito and the Google Jock team, who will stay remarkably restrained during the individual presentations, but once we get into the final panel at the end, which I hope will involve questions from all of you as we sum up this day, we'll have a lot of their synaptic firings exhibited on screen right here. And now the final installment of my little exploration of how biology and particularly the biology of my face is inspired by some machine design on the front of some famous automobiles. Here.
1948 DeSoto. Oh, man. Ooh, that's a hard one. 70 Dodge Coronet. Volkswagen, but it's just me. Everyone looks like a Volkswagen. A city car. Oh man. Actually, that's easy. MIT Media Lab city car.
Thanks to Paola Aguilera and the fine AV team here at the Media Lab. And of course, an applause for the great work of the CityCar team. We heard some of that discussion yesterday at the research reporting that was done by Kent Larson's group. But the CityCar has certainly been an inspiration to me and is one example of how the lab embraces big ideas and fuses technology with very, very complicated biologically based systems, and transportation systems certainly meet that particular test. Finally, as we prepared for this event and envisioned how we would involve a community not only in this room and among the sponsor network and friends of the lab and faculty and students that constitute this Media Lab community, we reached out to people across the world to invite them to participate, and hopefully they are watching and participating even now. One of the things that we did was offer a contest as an inducement for people to spend a little more time on our Facebook page. And what we did was something that the lab is actually very rich with, opportunities to take pictures of incomprehensible objects. You know, this is the planet of what the hell is that thing? Kind of, and if you walk around the lab, you can have that experience on virtually every workstation table. What is that? Where did that come from? Is that a sensor? Is that a piece of clothing? What is that? Is that a computer? Exactly. So we put 4 objects up on our Facebook page and asked people to explain what the objects are and then to tell us how they are related. And the winners, of course, were the ones that got it right. And I'm here to announce the winners, and there will be a prize. But first, the winners. Could I have the slide? Pair 1, Paco Gonzalez Blanche. Congratulations. And I can't tell you how these are related, and I'm not even going to try. Pair 2 winner, Christian Irvin. Pair 3 winner, Andrew Hollenbach. That's a little of Hugh, Herr's work there that he'll talk about in a moment. And pair 4 winner, Faraz Ismail. So congratulations to them. Little piece of as yet undetermined swag is in the mail and will be available to all of those winners. And it was so successful, in fact, that we're undoubtedly going to do that again at a date to be determined, but at some point, a very, very successful event online. Again, recognizing and emphasizing that the Media Lab not only is an institution for research, but is a community of people who are curious about the kinds of questions and challenges that the scientists in this lab confront every single day. So we begin in this last session, which is going to be very, very exciting, focusing on biological systems in a more literal way. And to begin, a member of the Media Lab faculty and one of the most exciting scientists and practitioners here at the Media Lab, please welcome Joseph Jacobson.
Thank you. Great. Thanks very much. It's a great pleasure and honor to be here, and thanks to all the organizers. If we can get the first slide. So what we want to talk to you about today is a field that a number of us are engaged in, both colleagues here at MIT and elsewhere, and you'll hear later in this session of one of the most prominent practitioners in this area. But it's an area we call digital biology, and the subtext is really the next Moore's Law technology. So I want to just start with a kind of an opening warm-up problem for you. Next slide, if we can, or I've got that here. So if you take a look on the top, there's a really interesting question, which is what governs the fundamental cost of doing things, of making things? And so if you look on the far left, we've got a microprocessor made by Intel, a quad Tukwila, about 2 billion transistors on it, a cost of manufacturing about $50. You've got a flash memory that's got the same 2 billion transistors in it, same 34 mask layers in it. The cost of that is about $3 to manufacture. And if you look at the raw materials that go into that, the silicon wafer, the area sufficient to hold those 2 billion transistors, you're well under $1. Even more telling kind of example is an area that we've been kind of deeply involved in here at the Media Lab, is kind of the cost of information devices. So if you look at call it a netbook, where you have a cost of $200, if you look at the raw ingredients that go into that, the costs of those are almost negligible. So what are the real fundamental drivers? What sets the kind of fundamental limits of how much complexity can we give to you kind of per unit cost? So this is a metric that we really like to think about and use. I'm not going to go through all the math, obviously, but this is a metric we call complexity per unit cost. And the basic idea is, if you give me $1, how much complexity can I give back to you? What's the fundamental driver behind that? And this is such a powerful metric to employ and to think about, because if you control that metric, you can really deliver things and make things that other people can't do. Or in another way, you can make things and deliver things to much broader sets of people than is possible if you don't have control of this metric. So just a kind of one way that we've used this, for instance, is when we look at the complexity per unit cost of things like wafers and transistors. So if you look at normal semiconductors, normal wafers that are made for microprocessors, Those take about 3 weeks of 7x24 processing, incredibly process intensive. We kind of use this metric to ask, what are the manufacturing technologies? What are the technologies that we have that are the extreme in this metric of complexity per unit cost? And you immediately come to an idea that's now been termed, that we've termed printed electronics, and you can start to do things like this. So this is a wafer. This we're making in a fab on the West Coast, about a 60,000 square foot fab. So these are circuits that are made not by normal silicon, but these are made by printing. So instead of 3 weeks of 7x24 processing, you can now make stuff in a couple of minutes that kind of hits the extremes of complexity per unit cost and starts to get you to places that end in things like $10 tablets and e-books and things like that. So when we started thinking about this, we started asking the question, well, what's the kind of extreme of that? What's the furthest examples that we know of in this metric of complexity per unit cost? And it turns out to be biology. There's nothing that is more extreme, not just by factors of 10 or 20, but by many, many orders of magnitude. I'll just give you a very quick example. As some people may know, inside of all of us is a set of machinery. It's machinery that is built out of proteins, and that machinery is sufficient or successful in being able to copy all the information that codes for us. So if you think about the human genome, and of course we have the gentleman here who first sequenced that, you've got on the order of 3 billion bases. The piece of machinery that copies that is a piece of machinery called polymerase. And it can zip along a piece of DNA at an extraordinarily high rate, on the order of 1,000 bases a second it can copy. And it can copy those with a precision that's extremely good. Only once in a million or once in 10 million times does it make an error. And if you compare that to the best that we know how to do as engineers, as chemical engineers or chemists, it's just extraordinary. So if you look on the left, is an extremely impressive piece of chemistry. This is the chemistry that we use to artificially synthesize DNA. It's called protection group chemistry, and it's the most precise chemistry that we know of in all of organic chemistry. But if you look at the metrics on it, it's vastly different from what biology knows how to do. So if you look both at the error rate, it makes an error which is extremely good as organic chemistry goes, but it makes an error 1% of the time. So 1 time out of 100 or so, it will make an error. And then in terms of the number of additions that it can make, instead of adding 1,000 things a second, it takes about 300 seconds to make each addition. And so if you look at the difference in this complexity per unit cost space that is possible within biology as compared to the best that we know how to do as engineers or chemical engineers, it's just extraordinary. It's about 8 orders of magnitude, about a factor of 10 to the 8. And when you see something like that, that's something that you just really get excited about and want to go after. So what are the things that really have us so excited? So if you kind of just take a quick snapshot of what is digital biology, what is this area that has all of us so excited, the basic idea is actually very similar to silicon in a certain way. So in silicon, we'll look at an example in a second. You have a microprocessor. You can download a new program into that, and you can program a completely new application. The same is true in a very formal sense with biology. So in biology, I can start with a set of code on the far left. That code is now not ones and zeros, but it's an alphabet of 4, A, G, T, and C's. Those are the nucleotides that make up DNA. I can code those into physical genes, actual genes. I can string a set of those together to make something called pathways, which are sets of genes. And I can use that to load a new program up on the top into, for instance, a bacteria to now make new chemicals, new pharmaceuticals. The number of new areas that are addressable just in the top rung of this is extremely vast. There's something like $2 trillion of commodity chemical in terms of a market size, and the vast bulk of that is addressable by digital biology or by synthetic biology. That is just kind of the beginning of where the imagination goes. if you start to look at a number of other applications, you start to get to places where we start to load programs not just into cells that live outside of us, but to cells that live inside of us. So there's a fantastic program that first kicked off with the NSF called SynBurk. And the idea there is that all of us have more bacterial cells that live in us than our own cells. What if we could recode those, load new programs into those so that we can code them to have early detectors for cancer markers, for instance, something that's monitoring us 7/24. Ultimately, is the prospect of actually being able to recode our own DNA. And we may want to do this for a whole host of reasons that have to do with disease and other things. If you look at this, the richness of this space, it's just extraordinary. So the question is, how do we get to that? So this is just a kind of a blow-up of kind of how this process takes place. We start very much like we do in the silicon space with what we call a parts library. It's a set of parts that can code for sensors, can code for logic elements. We can string those together into a program that we call a pathway, and then we can load that program into a cell of our choice, a bacteria or a yeast, and use that to produce, for instance, a new pharmaceutical or a new chemical. So the only problem with that is that as compared to silicon, we're in the very, very early days of that. So this is kind of how all of us think about silicon. We go into a design environment. We can pick out thousands of cores, meaning logic elements, a microprocessor, a DSP, all of the elements that I need to go into my cell phone. I go into a fab. A few weeks later, I've got my working chip. Chip goes into product, and I ship that. And more than that, I have a very fluid marketplace that all of those parts can come together. Where are we in digital biology or in synthetic biology? Well, we're kind of here. So those that can remember, there was a period of time where we had silicon transistors, but they were all discrete transistors. And this is in the days before the integrated circuit. And you had vast rooms of people that assembled these transistors into circuits. And there was a great term of art at that time called the tyranny of numbers. And what the tyranny of numbers was that there were so many great ideas of different circuits, devices, things that people wanted to make. But you know what? If they had more than 1,000 transistors in them, they were impossible to make because the yield on putting 1,000 transistors together, wiring up 1,000 transistors, is almost zero. So you had an incredibly exciting kind of prospect of electronics. You had transistors which were smaller than vacuum tubes, but essentially you had a linear kind of industry where each addition in capacity or each addition in complexity was essentially linear. You had to put in a linear resource to get a linear expansion in that. Along comes the integrated circuit, and all of a sudden we have Moore's law. So the place that we really need to get to make synthetic biology as exciting as we all believe that it is to create a fab for biology. And so the way that we're doing that is the following. There's a really nice set of technologies that have been developed by a number of different groups that works in a couple of different ways, but ultimately it allows you to take a real chip, a chip that looks like this, and come in and program small pieces of DNA to exist on or to be synthesized on that surface. And the reason that that's so powerful is that now, instead of having one machine that makes one piece of DNA at a time or small numbers of pieces of DNA at a time, now in a single shot, I can make enough DNA to code for 100 organisms, for instance. So if you just kind of run the math on that, the chips that we're using, for instance, to make our genes on a single chip has the output that now requires companies of hundreds and hundreds of people to synthesize. It's an incredibly high degree of leverage. So you can throw in some additional technologies to be able to put these pieces of DNA together and to be able to create genes. And you get to something like this. So the basic idea is you start with the set of pathways, the program that you want to make. You program that into a chip. There's a whole bunch of other steps that have to take place in order to make that DNA of a high enough quality to come off of that. And then we do a assembly process where we can take the DNA that comes off of that chip and assemble them up into pathways that we then can put into cells. So let me just end with a couple of applications, a couple of different things where we're starting to aim this technology. So one area that's really exciting is that we actually have a ton of content, a ton of stuff that we want to go build that we've just been unable to make so far. So to give you one example of that, if you look into GenBank, the number of bases there that code for content, things that we might want to make, things like enzymes, is extremely vast. So there's over 10 to the 12 bases of genes, things that we would like to go build. The problem is that the total synthetic capacity on the planet right now is only a very, very small fraction of that. It's less than 1/1000th of the capacity. And remember, this big circle here is growing at an exponential rate. It has a Moore's law that's even faster than the silicon Moore's law. So just to give you an example, to take the enzymes that are on the market right now, the top 1,000 enzymes, if we want to go build all the homologs that are in GenBank right now, that would be a project that would exceed several $1 billion to go do with the current infrastructure for making genes. But coming from chips, these are the kinds of projects. And in fact, we have a large-scale project to do just this. And then I'll just end with a last slide. And this kind of points to kind of future directions where we want to head. And you're going to hear more about this in just a little bit later in terms of these directions. But a really interesting thing is that if you look at any organism that exists on the planet, from a bacteria to a corn to a person to a cow, they have exactly the same genetic code. It's a striking feature of biology, but it need not be that way. That genetic code was a choice that nature made, but it need not be that way. And just to give you an example, we all know to get our kind of essential nutrition, we need on the order of 20 amino acids. Biology builds, it chose to build with 20 amino acids. It codes for a coding space of 63, 64 amino acids. It could build with more, but it chose to build with a particular 20 amino acids. We have catalogs of more than 2,000 amino acids of things that we would like to build with, we might want to build with. We'd like to be able to make organisms that can code that have totally different genetic codes. Prior to very recently, that constitutes just a massive kind of engineering undertaking. The number of bases and the number of things that you have to synthesize really is a massive multi-million-dollar task. But again, coming from chips, these are the kinds of things that we can do in kind of two-day kind of timescales. So I'm going to end there and pass to the next speaker. Thank you very much.
A couple of questions just to illuminate the process just for a second. Do you anticipate that this DNA production with chips will be a fully automated process, and in which case, where do your outputs go? Are they stored? Do they immediately go into organisms and then become part of this manufacturing of actual output chemicals?
Yeah, no, that's a great question. So at the end of the day, you really want the answer. So the output from this will go into organisms. Those organisms will express different versions of the different programs, just like you never get the right program right the first time, or even when you're making an ASIC for a new product, it's never right the first time. You've got to spin that a number of times. It'll go into different organisms. You'll try out different programs, and you'll capture the winner, and that'll be the output.
What's the most exciting, clinically applicable prospect from this technology, either in terms of a therapy or the delivery of some chemical that's very, very difficult to obtain by any other means?
Yeah, so I think one of the great prospects is there's a whole bunch of natural products that can be made. They're typically expressed in very low, slow-growing organisms, things like trees. We'd like to capture those, put them into very fast-growing organisms. Although the genetic code is the same, the dialect that they speak is different. So you have 64 codons that code for 20 amino acids. There's redundancy, and the particular choices that are made are different for different organisms. And so we need to recode many of those to get to kind of the right answer. So those are kind of early applications, I think.
Finally, you talked about the possibility of exploring perhaps some level of suspected arbitrariness in the way that for nutrition, for instance, it's 20 amino acids that the genome codes for. It could be up to 63. You express some sort of set of limits. Given that on some level it appears that this whole synthetic digital biology movement is a way of ripping off a previously existing DNA code and trying to develop applications. Right. What is the level of complexity to move from using that pre-existing code to really exploring the parameters of what is a sufficient condition to produce complexity that would simulate life? How few amino acids? What are the parameters?
Right. No, it's a fantastic question. And the limit to us has been a limit on learning. So in the semiconductor world, since you've got a Moore's Law kind of capacity, you can make literally thousands of circuits. And so when Intel wants to go from a 32 nanometer to 28 nanometer with a known working microprocessor, they still have to make thousands of different versions of that to find ones that work. Then they can update the CAD rule set. We've only made on the order of 10 working pathways that have gone commercial. And so somewhere towards the end of this year, we're on schedule to have in a single cluster tool more capacity than exists anywhere else on the planet for synthetic genes in one cluster tool. And we think that's going to give us the tools to go make things at such a high capacity that we'll be able to extract the rule sets for those and then go apply those in an engineering way. So.
Only one IP question here. You develop a chip that makes a DNA instruction set, which then goes into a yeast, which makes a molecule that treats epilepsy. The yeast made the molecule. The yeast does not get the patent. What along this pathway is patentable and ownable, and how are those IP restrictions an inhibitor on innovation in the field?
Well, look, I think it's very, very early days in this field, and the whole goal of being able to do this is to stand up a capability just like we have in the semiconductor space, where you just have a lot of kind of cross-sharing and cross-fertilization. I think what we want to do is really break down the barriers that exist right now in pharma, where typically you're developing something in silos. There's very little cross-flow. If you look at the semiconductor space, you go develop a new chip for a cell phone, that's got cores that come from IBM, from TI, from university, all on one chip. That's the point that we want to get to. So,
Joe, thanks so much.
Thank you.
Let me tell you a slightly personal story about our next presenter. I've known Hugh Herr for quite a long time, and when I first met him, we were bonded because we both share this vaguely comparable disability space. I have a spinal cord injury, and Hugh is a double amputee. And when I first met Hugh, the technology of my wheelchair was roughly comparable to the technology of his prosthetics. I mean, they were pretty cool legs that he was using. He had some ideas about some future technological upgrades and that sort of thing. All very, very exciting work. But in a sense, he and I were both sort of having a collaboration with a machine that helped to sustain our lives. In my case, a wheelchair. In his case, whatever prosthetics he was using. Almost immediately, our paths diverged. And while the technology of my pathetic, if useful, wheelchair has remained the same, in Hugh's case, the technology that he collaborates with has gotten better and better and more exciting. And so while we're roughly in the same space, Hugh occupies a stratospherian level of collaboration with technology that is exciting to behold as another disabled person, and really points the way for the kinds of collaborations that will produce productive, tangible quality of life upgrades for millions and millions of people. Please welcome member of the faculty here at the MIT Media Lab, Hugh.
Thank you, John. I mentioned in the preamble to this symposium that in the future, there'll be an intimacy between the body, the mind, and technology, synthetics. We'll embody technology, and technology will embody us. That's already starting to happen in the field of bionics. So bionics is defined as using electromechanical elements that are attached to the body or implanted inside the body that normalize or extend capability, physiological capability or cognitive capability. So what we're starting to see in this field is, as I stated, embodiment. What do I mean by that? A person that's fitted with a bionic device that has some leg pathology or arm or whatnot. If it's truly a bionic technology in that it emulates normal physiological function, it's not a tool to that person. It's not a separate lifeless device. It's an integration that they have with the technology. It's embodiment. Key to that level of intimacy is the science of extreme interfaces. So I've defined 3 extreme interfaces between machines and the human body. One is dynamic. In the case of bionic limbs, can you architect a limb that moves dynamically like a normal biological limb? The second is electrical. Can you have electrons flowing from the human body, the nervous system, to the device, and then from the device back to the human? And thirdly, mechanical. How do you attach machines to the body mechanically? So my talk, I'm going to go through these extreme interfaces, talk about work that's underway by my group and my colleagues. And then I'll finish up with a note on clinical impact. So I'll begin with dynamic extreme interface. So as I mentioned in my preamble, years ago with tissue engineer Bob Dennis, we decided to build a hybrid device, a robot that swims, that's actuated by living muscle tissue, skeletal muscle. We wanted to do this exploration to really think about, with artificial stimulation of the muscle tissue, could we in fact control machine movements in a hybrid device? And we found that we could, but the dominant issue, of course, was organ maintenance. We couldn't keep this machine alive for more than 48 hours. But we learned a lot about control. So you can see the swimming looks quite natural. So we applied, again, the actual structure, the actual tissues, and a control that resembles skeletal muscle control. So I thought to myself, well, it's going to be a long time until we understand, science understands organ maintenance, how to either cell culture or extract tissues from a body and keep those tissues alive for an extended period of time. Can we think about when we control synthetic devices, to control them in a way where the device moves in a natural biological way? So here's a schematic of the leg, and then a structural model representation of the leg showing the muscles and the various tendons of the leg. So what we've done is, in fact, in my group, is we've advanced a mathematical description of the body, what the body does in standing, walking, and running. So here's an animation. When these balls expand and contract, that's when the muscles, the biophysical muscle models, are being activated. So what we've learned from this model is nature doesn't have disciplinary silos. You know, we often think of mechanical engineering in one silo, electrical and computer science, and so on and so forth. Nature, there's this gorgeous integration between structure, morphology, and neural control in the case of legs and locomotory function. One can look at the calf muscle and the Achilles tendon, and just by looking at its structure, you can say a great deal on how it's controlled. And by looking at how it's controlled, you can say a great deal about its structure. So that integration, that intelligence in the wiring itself, allows for extraordinary function and locomotion. So in this model that I've just shown, it's highly reflexive. It's a spinal animal, a spinal model. So what we do is we take joint state, positions and speeds, and we feed that back into a hypothesis in terms of what the spinal level reflex is. So here we see a feedback that poses a force and position and speed feedback, which then dictates the neural activation of the muscle. So what we can do in machine control, we can put forth a biophysical-based controller where we take sensory information of the robotic joint's position and speed. We feed that through the biophysical model, and the biophysical model tells us how the joint would move if it were made of flesh and bone, muscles and tendons and so forth. So the bionic limb can be made of titanium and silicon and carbon, but it can move naturally and integrate with the human. So just one example, here's a foot-ankle bionic device that we've been developing in the last several years, and I'm wearing 2 of them. This device is the first of its kind to actually normalize locomotory function in persons with leg amputation. So these data shown here quantitatively show that normalization. So here in plot A is the ankle angle when the foot comes off the ground and walking versus walking speed. The blue symbols are the bionic device, and we see the dashed line is the mean value for a person with biological legs. And here's work, the energy output normalized by body mass in kilograms versus walking speed. You see the bionic device is actually doing excessive energy at low speeds. And here it just shows as a person walks faster and faster, they get more and more energy. Because of these normalized mechanics, for the first time, we've normalized how much food energy and the metabolism of the leg amputee as they walk. We've also normalized walking speed. So what I'm going to show you now is a video. This is a US soldier that lost a limb in battle. He was fitted with this device just an hour previous to the video being shot. He was taken outside, walked outside, just decided to run because he was inspired. So what's fun about this is that by extracting principles of what's happening in the human body when you walk, it turns out those principles also allow for running. That is an emergent response of the basics of walking. We also got running as a freebie. What's also wonderful about this is the short amount of time that the human, the biological part, adapts to the synthetic structure. Because we've carefully modeled the synthetic bionic device, and it has similar dynamics to the biological limb in that section of the body, the amputee's body is used to those dynamics. So we often fit patients with a bionic limb, and in 3 minutes, they've adapted. It happens very, very rapidly. So I'm going to talk about the second extreme interface, electrical. So obviously, when a limb is amputated, key nerves are amputated. They're transected. And what we want to be able to do in the future is communicate to those transected nerves. So what we're exploring is how, in fact, that might work. So our strategy is to take target end organs, tissues that the nerve likes to connect to, such as skin cells, muscle cells. And we're putting those in close proximity to the amputated nerve. And what the nerve does, in fact, is grows again, regenerates, and attaches to those end organs. So there's an image. There's a stack of literature, a great deal of literature that shows if you have a muscle, shown here, and you put that muscle is a denervated muscle, and you put a nerve next to that muscle, the nerve will sprout and attach to the muscle. It's very repeatable. So we've been doing experiments, first with Inercy Technology, a local company, and now here in the lab, it's directed by research scientist Ron Rizzo, in a rabbit animal model where we put skin cells and muscle cells next to the amputated nerve. There's sprouting, there's attachment to those end organs. Then the body of the animal quickly vascularizes the tissue, and you end up with a completely stable, viable implant. So what we want to do is extract the motor signal from the muscle sliver, because muscle is a natural amplifier of the signal in the nerve. And we also want to eventually stimulate through the cutaneous axons, because we're interested one day of taking sensory information from the bionic limb and reflecting it on the nervous system, so that a person could perhaps feel their prosthetic device. So we're also interested, of course, in getting signal from the muscles that still are present in the person's leg. This is a technique that's been used for decades by measuring the electromyographic signal. When your spinal cord activates your muscle fires. That can be recorded and sent to the machine. So what are we doing with these signals? So back to this modeled spinal level reflex. We're discovering that if you use the neural signal to adjust the gains, the sensitivities of the neural reflexes, the amputee goes from being a backseat driver to driving the car. They actually have direct control over their bionic limb. So here's a plot of power versus showing the tracking under neural control. So what we're doing is this interplay between the intrinsic bionic prosthesis and the extrinsic signal, or the neural signal, where the very rapid loops are taken care of by the intrinsic device, the bionic device, and the very slow, low information signals from the neural commands are used to adjust the sensitivities of those reflexes. So let me go on to the last interface, mechanical, and I'll talk about clinical impact. So how do we attach machines to the human body? Believe it or not, it's not well understood. If you take 1,000 people that are using prostheses and also braces, like a knee brace, and you ask them, what's the #1 problem you want solved? Probably all 1,000 will say, it hurts. You need to fix the fact that it hurts. So what we're thinking about here is really expanding dramatically the data that's collected on the human, really characterizing the part of the body that you wish to attach the robot to. So Andrew Malinsky in my lab, a graduate student, has said, well, let's look at how the skin stretches when you flex and extend a joint. So here's a knee joint that's flexed, and you see the red areas is where the skin around the patella is being stretched, where it has a great deal of strain. And what we want to do with this information eventually is the synthetic part that directly interfaces with the skin, we want to move in the same way that the skin is moving. We're also looking at MRI data, which gives us the shape of the residual limb, as well as information that we can input into a biomechanical and anatomical model that tells us how stiff the body is. So on the right is this mapping of how stiff the residual limb is. This is a transtibial amputation. So in the red regions, there's a lot of tissue depth, soft tissue depth, so the person's soft in those regions. The green areas where there's skin and bones, they're very, very stiff. What we're then doing is doing a mapping from how stiff the body is to a 3D printed interface where you have a spatial variation in the stiffness of the interface that's directly quantitatively computed from the MRI data. And we've already shown with these novel sockets that the peak pressures are lower. So the conventional sockets in red and the new sockets in blue. So very large reductions in peak pressure as a person walks. So in the future, we not only want to vary parameters spatially, but also temporally. So here's a material that's being advanced by SRI International, our collaborator. You apply a voltage, it becomes stiff. So now in these interfaces, if the person sits down with the knee brace will just relax. Because there'll be computational and sensing. The device will know the person's sitting, and it'll just relax to make it even more comfortable. So on being bionic, a person that receives a bionic device, again, does not view that device as a tool, like a hammer is a tool. It's embodiment. It's full integration. So I'm going to give you two case studies now of persons that have been recently fitted with a bionic device. The first is Ed. Ed's gone through, I think, 40 surgeries in his leg. He was in a motorcycle accident, and he was recently fit, and he had this to say: having the bionic leg has given me back my life. I feel whole again. and integration. And here's Ed's story.
My name is Ed Lastowski. In 1967, I joined the Army, and I was medically retired in 1984 after having a motorcycle accident on the 7th of August, 1981, where I took off most of my thigh, broke my femur, severed the artery in my right arm. I had 39 operations between all the hospitals, and West Roxbury was the last one, and that was number 40. And that was the amputation. I feel more like a human being, complete, whereas, I can watch people in the eye as I walk down the street instead of watching the ground or where I'm stepping. It's, being a normal person again.
So Ed has reduced his pain meds by 1/3 as a result of the bionic limb. So the stress throughout his biological joints has been dramatically lowered. A final case study is Stefan, living in Mississippi. And just the emotional impact here is remarkable.
These are tears of joy.
How was that?
Great.
Good.
I haven't walked on a movable ankle in 14 years.
Thank you.
That's great, Hugh. Let's explore this embodiment idea a little bit in those particular case studies that you mentioned. You talk about how the signal and all the qualities of motor motion are a combination of neural impulses and something about the design of the tissue itself that contains within it the instruction set, call it what you will, that produces the natural seeming gait and other sort of physical motions. What do you think is going on in the brain when either you've had this experience or some of your subjects have had the experience of this adaptation where an intuitive designed prosthetic seems to marry itself with some set of expectations in the brain that makes it feel, and as you've testified here, to the users as though they actually have a leg, not a tool, not a new prosthetic, not a crutch, but an actual leg. What's going on in the brain, do you think?
Yeah, the case studies I show, the individuals were not born with the missing limb. They lost their limbs later in life. And so there's some memory there. So when we put the device on, the normalized dynamics, those forces and positions which are felt throughout the person's biological bodies are normalized. And I think it would be a very interesting study after months and months and months of use of this device that emulates to look at their brain scan before and after and to see if their brain has become more normalized in the motor
This adaptation
areas.
process you suspect is reactivating some sort of center of motor memory.
I would expect so, yes.
You talked about how in this one extreme interface that you mentioned, there would be a transected nerve that would connect to an electrical implant that then vascularizes, I guess that means grows blood vessels, et cetera, and becomes a kind of hybrid interaction between an electronic device and a neural impulse.
Right.
The assumption there, though, is that the motor signal coming off the transected nerve contains all of the signal required for walking, when you've just said embodiment means that the signal is all over the place. Why is that transected nerve so important?
Well, so in my case, as an example, my legs are amputated here.
Right.
Obviously, I still have musculature intact above my knee. Also, obviously, the muscles that used to be in my feet are no longer there, but the nerves, the transected nerves, are still there that used to feed those muscles. So not only do we have to tap into the muscles that I already have, but we also have to tap into the nerves to get the full motor intent and also afferent feedback.
But do you believe that there's more than just a trigger in that signal that's coming down the transected nerve? Okay. Yeah. What's the evidence of that? That there's a lot there other than just an on-off?
Our biophysical models, you know, we can show that to do simple variations like from walking from slow to fast, that the stiffness of the muscle, for example, is being modulated. So it's just not a bang-bang on-off binary control.
Finally, how much harder is the problem of taking some of these processes, both in terms of interface and in terms of physical embodiment, in developing arm and hand prosthetics?
Yeah, arms and legs are both difficult, but they're difficult for different reasons. Legs are hard because of the tremendous loads and powers that they have to resist and also create. But because in legs, the robot is sandwiched between the human and a surface, the ground, you can do a lot of stuff intrinsically with sensors on the device itself. So there's not I would say, a profound need for the neural command in legs as there is in arms. In arms, when I decide to reach out and grab my coffee mug, that's direct motor intent. And I can't do an interaction sensing with a surface because a surface doesn't exist.
Do you think the feedbacks in the extreme interface that you're talking about with legs will eventually have applications to create enough of a feedback signal?
Yeah. All over the body.
All right.
And I want to dance really well, so that's why I want that feedback.
You want to dance really well, or you do dance
well, I
really
never
well?
could dance well, so that would be...
So you became an amputee so you could dance well?
Yeah, exactly.
Nice. That's MIT Media Lab for you right there. Hugh, thank you so much. It's a very exciting moment now. Certainly everyone would agree here and in the scientific community that the sequencing of the human genome was an extraordinary moment in human history and in the history of science. What perhaps was not anticipated was that it was also a door leading to a much more complex and rich level of exploration of genetics and the sort of instruction code of what we think of as life that is even beyond what was anticipated to be the power of the human genome itself. That work is continuing, and we've talked a lot about it over the course of this day and a little bit yesterday. J. Craig Venter, of course, is responsible for the sequencing of the human genome, also responsible for reporting the synthesis of an actual biological organism using techniques at the two firms that he founded, the J. Craig Venter Institute, of course, and Synthetic Genomics, his two companies. J. Craig Venter is an extraordinary pioneer and a holder of milestones in this understanding of identity, biology, and design, and using technological tools to advance our exploration beyond where we might have ever imagined going even a generation ago. Please welcome, join me in welcoming Dr. J. Craig Venter.
Well, thank you for the extremely warm introduction. That's a difficult one to maybe live up to, but it's also a pleasure to be here at the Media Lab. I've been a long-term fan, and it's also interesting to be at a place that's inspired by biology. We go the opposite way. We use computer metaphors to explain what we're doing in biology. So our biology is inspired by what you do. So I'm going to just show you some of the early steps that we've taken in this journey from reading to writing the genetic code. And but first, we're actually putting some of our designs into a larger scale. So this is the Venter Institute, which is a not-for-profit institute. The new building that's under construction at the UCSD campus in La Jolla overlooking the ocean. And we've tried to put some of the design to create what I think is going to be the first carbon-neutral research building containing large banks of computers and all kinds of wet labs. And this will be finished next October. We're actually using some of our biological conversion processes to convert wastewater into drinking water if you want to join us for a drink sometime to test those principles. So I'm going to talk about what you've heard a lot about already, and that's the biological world and the digital world. I view DNA as an analog molecule. And it's a code, as everybody knows, with 4 bases. And what we've been doing starting extensively in 1977, when Sanger's team read the first bacterial viral genome, we've been digitizing that biology. So we go from this 4-letter code to the 1s and 0s. And through this journey, everybody takes into account all these assumptions that this has represented biology. There's obviously been some debate about it. But we've decided for a number of reasons to see if we could go the other way. Could we start with the digital world and recapitulate biology? So some of the early readings, the first genome of a living species our team did in 1995, and then 5 years later, the first draft of the human genome, as you heard a few minutes ago. But in 1995, we did a second genome. We chose the genome of the smallest organism with a self-replicating system, and that's Mycoplasma genitalium. And as soon as we had the first 2 genomes in history, we started asking questions about what was the minimal design of life, and could we eventually reconstruct a minimal gene set to understand the basic components as the basis of future design. We took a number of routes to getting there, and ultimately we decided that we had to actually build the entire chromosome chemically because of the limitation of biological systems for redoing reductions. And as soon as we made that decision, then we had all kinds of new questions. Would chemistry even allow us to build entire bacterial chromosomes? And if we could build them, could we boot them up? Or would we just have a really large piece of DNA? So we set up a couple of teams to go down this route. As you heard earlier, DNA synthesis is kind of a crude art right now. It's not very accurate. It's an N-1 situation. So the longer you make the piece of DNA from a synthesizer, the more errors there are. So we set out early on to find ways to correct those errors and improve the quality of synthesis. And our test system was Phi-X174, which was the virus that Sanger sequenced in 1977. And we started with the digital code in the computer and 4 bottles of chemicals and reconstructed the 5,000 base pair genome. The exciting phase came next when we inserted it into E. coli. E. coli recognized the synthetic DNA as normal DNA, started reading it within seconds of it going in the cell, started making all the viral proteins. The viral proteins self-assembled, formed the virus, and the virus showed its gratitude by killing the cells that made it. So there's all kinds of metaphors, usually in biotechnology, for this, but we call this a situation where the software is building its own hardware. All we did was put in a set of software instructions, and we ended up with this viral particle that had activities in conjunction with a living system. Now, our goal was not to make viruses. That's pretty easy to do now. We wanted to make something much larger, but we figured if we could make viral-sized pieces of DNA accurately, we could string a whole lot of these together and make an entire chromosome. So that's what we set out to do. So we synthesized a large number of viral-sized pieces, and we started combining these together. And this was very slow and laborious when this started out. We would clone the pieces in E. coli. We would sequence them to make sure that what we put together was accurate and stable. And then we went on to the next stage. So we went from 24 kb to 72 kb to 144,000 letters. At the time, the largest piece of DNA that had been made was only 30,000 letters. And that was a process that took a very long time for a group to do. The trouble is, once we got over 100,000 letters of genetic code, it was not easy to grow these pieces anymore in E. coli. So we looked around for a new system, and it turned out brewer's yeast was the ideal system. It could take in large pieces of DNA. And there's a thing in biology called homologous recombination. When there's overlapping pieces of identity, yeast is able to assemble those pieces together. So after synthesizing 4 quarter molecules of the genome with the right overlaps, we essentially just put these pieces into yeast with a small piece of synthetic DNA, a yeast centromere. And just adding this eukaryotic centromere to a bacterial chromosome, it turned that bacterial chromosome into a eukaryotic chromosome and would grow and replicate in yeast. So this is what we published in 2008. This was the first complete synthesis of a bacterial chromosome. At the time, this was the largest molecule of a defined chemical structure ever made. Now, we were working continuously on the synthesis problem, and we had some nice young scientists working with us. One is Dan Gibson, who made a real breakthrough on what we were doing with the combination of these pieces of DNA. And he found that by putting the 3 enzymes we were using that basically trim back one strand so they join together and then you stitch them together, we could do this all in one tube at 50 degrees centigrade. And so this means now that it could be automated. And so we've been working for the last couple of years on the automation of these processes. So we can start just with the digital code in the computer and make very large pieces of DNA in a completely accurate form. And we now have new error correction methods that on top of improving the quality of synthesis, we can make very large molecules with 0 errors in them quite rapidly. The goal is actually to extend this. It took us a couple of years to make that first chromosome. The one we published with the first synthetic cell at 1.1 million letters, that I'll show you in a few minutes, it took about 3 weeks to do. We want to get so we can make literally millions of chromosomes a day. And the reason for that is we don't know much about biology. And unlike what exists in the electronics world, we have tens of millions of design components, most of which we have to learn what they do. So how do you boot up a synthetic chromosome? And so our biology team was working in parallel, and they came up with this study that I think is one of the most important ones for understanding the fundamentals of life and biology. And in this study, we simply replaced this DNA software in a cell with that from another cell, and it completely converted one species into another. And because of the importance of this for understanding the future of biology, particularly as we design new biology, let me walk you through this. So we isolated the chromosome from one species, M. mycoides, and we treated it harshly with proteinases because a key part of this at the time, we wanted to know if we could synthesize and use naked chemical DNA. And we added a couple of gene cassettes to it, one so we could select for the chromosome, and another where it would turn the cells bright blue if it was activated. And we found methods for inserting this in another related cell, roughly the same distance we are from mice, about 10% different. So here's a movie that would make the media lab green with envy. We inserted the chromosome into the cell. And so now we have a situation with the body of one species. And right now it has, at this moment, 2 sets of genetic instructions. As soon as you put this chromosome in the cell, it starts being read within fractions of a second. And so it started reading this chromosome, and new proteins were being made. And some of these proteins are the restriction enzymes that my colleague in all this work, Ham Smith, got the Nobel Prize for in 1978. These are the enzymes that cut DNA at very precise points. These enzymes recognize the chromosome in the cell as foreign DNA and chewed it up. So now we have the body of one species and the genetic instructions of the other. So what happened? In a very short period of time, we ended up with these bright blue cells. And when we interrogated them, there was not a single molecule of the original species in these cells. Every protein in the cell, every molecule derived either from the new proteins that were synthesized based on the new software we put in the cell, or derived directly from those proteins. So this is why we call it a synthetic cell. Everything derives from the new DNA instructions. So if you didn't understand it before, it's important to understand life is a DNA software system. And if you change the DNA software, you change the species. And this then becomes the principle of design as we can work with writing genetic code. There's a longer, complicated story because some of you might have noticed we're actually assembling the bacterial chromosomes in a eukaryotic cell. So we had to find a way to get those chromosomes out of the eukaryotic cell and put them back, transplant them back into a bacterial cell. And so we developed a method for cloning a whole range of bacterial chromosomes as eukaryotic chromosomes. And it's as simple as adding the synthetic yeast centromere to the bacterial chromosome, and then it grows and gets carried along as a eukaryotic chromosome in the yeast cell. And when we first isolated the mycoides genome that we were growing in yeast and did the transplants, they didn't work. And so much of science is problem solving. It's not just creativity of new ideas. And it took us 2.5 years to solve that problem. The fundamental issue is when we took the chromosome out of the native cell, it had been methylated. And bacteria use DNA methylation as a way of protecting from restriction enzymes. So when we took the unmethylated chromosome and transplanted it, the restriction enzymes in the cell with the native chromosome chewed it up, just like happened the opposite way in our first experiment. And we proved this by isolating all the genes for restriction endonucleases from the mycoides genome, and if we then methylated the DNA out of yeast, we could transplant it. And we proved this by altering the genome of the recipient cell, and if we removed all the restriction enzyme genes, then we could just use naked DNA out of yeast. So we had this process now with this complete circle that allows most bacterial species that have not been able to be worked on and changed very dramatically. We can clone the chromosome in yeast, we can make dramatic changes in it, isolate it, methylate it if necessary, and transplant it. So at this stage, we thought we'd solved everything and we were ready now to do all this with the synthetic chromosome. We decided because of the new chemistry, we'd build a much larger genome. So this was the 1.1 million base pair genome of mycoides. And we started with 1 kb pieces, put 10 of those together to make 10 kb pieces, put 10 of those together to make 100 kb pieces. And we took 11 of these 100,000 base pair pieces, put them in yeast with the right overlaps, and it assembled the whole chromosome. And we were so sure it was going to work, we did the transplantation, and it didn't work. And just like software engineers have debugging software, we had to create biological debugging software to find out what was wrong with our chromosome. And to make a long story short, we found a single base pair deletion in one gene that was essential. So 1 letter out of 1.1 million letters made the difference between life and no life. We corrected that one error, and we ended up with these, the very first synthetic cells just over 2 years ago. And the way we knew that these were totally synthetically derived is we had to invent new ways to watermark the DNA to prove that, in fact, it was synthetic and not some sort of recombination or natural DNA. And this was all published in Science in July of 2010. So to mark the DNA, we watermarked it, and to watermark it, we created a new code to write the English language in the genetic code with complete numbers and punctuation. And this had been done before using an ASCII code. In fact, one of the reviewers of our paper to Science sent the review back written totally in DNA code, much to the frustration of the science editors. But it was using an ASCII code, and the problem with ASCII codes in biology, it doesn't insert stop codons, and so you can get all new biological molecules, perhaps toxins, made. And that wasn't the goal of watermarking the DNA. So our code that's in the first watermark inserts frequent stop codons so you don't make new biology. Being the first organism with a computer as a parent, we thought it was important that it has a URL. And people that solved the code would send an email to the species saying that they'd solved it and were able to read it. And once you solved it, you'd be able to read the rest of this, and it has 46 names of the scientists that contributed to it. And with the first genome, when we made it, we just used the single amino acid letter code to write the names of scientists, and we were accused of not being very creative. There was no secret messages in it, nothing like one small step for bacteria. So for this genome, I came up with 3 quotations from the literature that I think everybody can read. The first one's from James Joyce, to live, to err, to fall, to triumph, to recreate life out of life. That seems somehow appropriate. The second is from American Prometheus, Oppenheimer's biography, after just hearing about Prometheus. See things not as they are, but as they might be. And the third from Richard Feynman, what I cannot build, I cannot understand. And once this code all became public, the first response that we got was a letter from James Joyce, a state attorney, saying that we didn't have permission to use this quotation, and they wanted some kind of damages. And we pointed out that James Joyce was dead. We couldn't really ask him. And the fair use laws allow you to use a paragraph or more without permission. So we dismissed that one. And then we started getting emails from a Caltech scientist saying we had misquoted Richard Feynman. And this is a dangerous part of the web because you'll find this quotation thousands and thousands of times over on the web. But to prove his point, he set a picture of Feynman's blackboard. And this was the original quotation: what I cannot create, I do not understand. And I think that's a much better quotation. One of his early biographers made a mistake, and that's why the other one's on the internet. So we've changed the genetic code so that it has the correct quotation, and so Feynman can rest peacefully. So I view these 60 million or so genes we've discovered to date, and we don't know what the final number is going to be. It's going to be somewhere between probably 1 and 300 million genes. This is what we have as a set of design components if we're going to write new biology. We don't need 64 additional amino acids. We sort of have an infinite design capacity, but those additional amino acids do have value in terms of creating viral resistant organisms, et cetera. So think of this versus just the few resistors and transistors, then integrated circuits. And so we have digital software in our computers at Synthetic Genomics for designing DNA software. And this is actually much more difficult than Joe made it sound. I mean, in principle, it is very simple. The trouble is, even with the smallest organism, the most minimal genome we know about, there's over 100 genes that are absolutely essential for life, and we have no idea what they do. So it's kind of hard to design things on first principles when we don't know yet the first principles of biology. We're in the process now we've designed 2 genomes that we're in the process of building the chromosomes for that, in fact, take all of our biological knowledge together to see if on some degree of first principle, including these 100 genes that we know if we knock them out, the cell dies, we're just putting those in gratis, and we're trying to design that. So we're in the earliest, crudest stages of this whole field. And what you heard earlier is correct. The thing that's going to make a huge difference is getting the cost and the accuracy of synthesis down. And we have some other ways to approach it. Chip synthesis is actually really inexpensive, but it's really inaccurate. And there's a lot of problems with getting selection out of it. But I think as it happened with DNA sequencing, there's going to be exponential changes in a very short period of time. Keep in mind the first version of the human genome was supposed to cost $3 billion and take 15 years. Our big breakthrough was reducing that to 9 months and $100 million. Now it takes 2 hours and a couple $1,000 within 10 years. So we expect to see these same kind of changes with at least a 1,000-fold difference in the synthesis cost. And that allows us to overcome the problems with our lack of biological knowledge. And if we could make 1 million chromosomes a day and just test them in a screening process, we can deal with all this unknown biology, in fact, have it be learned. And maybe this would be the place to help do it. We're trying to build a self-learning robot that can learn biology 10,000 times faster than an MIT student. Because we need to do that. At the rate biology is going, and with all these new genes, it would take centuries to understand what's there. So we're designing cells, trying to use sunlight through photosynthesis, use CO2 as the carbon source, and we're working down a number of avenues for fuel and food and chemicals. The first applications, and the fastest ones, are we now synthesize the flu virus as a new vaccine in less than 24 hours. So BARDA and the US government sends us a test pandemic sequence, and we have 24 hours or less to make that, and our teams have done it now in 12 hours. And we get that actually to Novartis here in Boston, who rescue it and have it ready to go into their viral synthesis production system for producing a flu vaccine. With photosynthesis, we're trying to change 200 parameters in cells to get them to do things that they haven't done before. But people have tried this for 50 years, and so we have to see if these new tools really give us a unique advantage for engineering much more complex systems than we have before. People worry about the ethics of creating new species and the implications of it. Before we did our first experiments, we asked for a complete ethical review that was done by the University of Pennsylvania, and the study was published in 1999 in Science. The Sloan Foundation has funded multiple programs between MIT and my institute and the Center for Strategic and International Studies in Washington. And you can download these reports from jcvi.org if you're interested. And when we announced the first synthetic cell, President Obama asked his new Bioethics Commission to take this on as their #1 charge, which they did and published this report in December 2010. And I think this is certainly one of the best reports in recent history out of any government bioethics commission that recognizes the tremendous promise of this new field, but also understands that it's a very powerful technology. And you don't need hundreds of people or thousands of people. You need 2 to 3 at the most to do design and synthesis and processing. So it changes what can be done. It changes how many people can do it. And it talks about making sure we build in biocontainment into the design of organisms and to develop a number of new biocontainment approaches. Thank you very much.
Sit down, Craig. Go ahead. Have a seat. So let's conclude this very exciting day by inviting Paola Antonelli, one of the most influential voices in design, Senior Curator of Design and Architecture at the Museum of Modern Art. We also want to activate the Google Jockey station up there, Joey and the gang. I suspect you have some things all pent up. Please spray them across the screens for us as we get started here. We'd love to see what's been popping off in your synapses over the last hour or so. First of all, Paola, welcome.
Thank you.
And I have a lot of questions, Craig, but I won't monopolize the discussion. First of all, your impressions of design as being an important motivator in the kinds of work that you've seen today. Do you see it here?
Oh, yeah. You know, I have a sort of a motto that's becoming a red thread in my career and in my life, which is that design is fundamental for progress because it takes scientific revolutions and innovations and transforms them into life. So there's a line that goes from, you know, the thinking of so many of the scholars here and the scientists here and people, and that's design. And you see it in the work of Hugh, and there's that last step from the innovation in technology to the real gait, to the real sense of having legs and feet and being able to walk again. You see it in the work of Neri's when aesthetics, the power of form, the power of color, the power of emotion, the power of mythology drives home the intention of new technology. So definitely design has this important role, which is that of being the second of the class, secondary but primary at the same time, because it's about making science and new technology into life.
Craig, you said there were tens of thousands of design elements in this mix of tools that are open to creating synthetic biology in the way that you've described here.
Tens of millions.
Tens of millions, okay. When you talk about design elements, are you talking about a parts list in the way that Ed Boyden might have discussed earlier today, in the sense that this does this widget does that? Are they apps in the sense that they're little pieces of software that relate to specific functions? Or are they more a palette of possibilities, and that when you think about synthetic organisms, you can imagine doing something better than nature does, doing something more efficiently than nature does? How do you think of design when you use that word?
I think in all the ways that you just described. I mean, we have everything from a single gene that you can put in if you want a very specific function. And the field's been very creative, actually getting equivalent of electronic gadgetry out of biology, of gating functions, oscillators, things that don't exist naturally in biology. Then we have complete circuitry. If you want a cell to metabolize sugar and generate the energy necessary for maintenance, that's one way to do it, or metabolizing hydrogen into methane, or metabolizing sulfur, whatever you want it to eat. Some of these can be designed cassettes for specific function. But that's what we're trying to get this core minimal genome, as this stuff is necessary for life. And everything we add to it, as has happened over 3.5 billion years, allows us to now take things in directions that natural evolution could never take it. For what we're trying to do with algae, we have to increase the efficiency by at least 10-fold over anything that's ever occurred in nature.
Which is similar to what Neri said before. You know, she said there's no pattern that ever is repeated in nature, but it took so long to get to those patterns. There are many similarities there.
Although, let me ask you a question about your synthetic bacterium. It sounds to me, and correct me if I'm lost here, you, in that compelling movie that you showed us, you inserted an instruction set that competed with the existing instruction set, and you modified the bacteria in a way that the restrictive enzymes that would have gotten rid of foreign instruction sets were no longer active, so that the introduced instruction set could take over the one that was residing there. Is that roughly?
That's roughly correct.
All right.
Yeah.
So then the instruction set began to be read off of your synthetic chromosome or gene or whatever you would describe it, the set of base pairs that you inserted. It replaced all of the proteins in that bacterium with instructions coded from your set, not from the original. My question is, this took place inside a scaffold, which was that organism. That organism was actually a scaffold. Now, you define synthetic life as being the result of your instruction set, not the one that was there. But in fact, everything that was in that bacterium was alive at the moment you inserted the instruction set, and during the entire time it was recoding itself, and then afterward, when it became, in your view, the venter organism. What is that that's going on? Is that not life, the scaffold where all this takes place?
Well, in fact, in Hugh's talk, he talked about having the muscle that was stimulated, but the muscle degenerated. In a typical bacterial cell, 10% of the proteins die every hour. And the reason we are DNA software systems is our systems need constant maintenance, constant turnover, constant replacement. So you can't have life with just a protein system and lipids.
So there was a clock ticking when you did your insertion.
As soon as you take a chromosome out of any cell, that cell will die. The longest living cells we have are our anucleated red blood cells, which roughly can last 2 months. But they have special structures to allow them to die slowly. A bacterial cell can die within seconds of taking out its chromosome. Other cells in our body have different rates of death once you remove the instructions. You can't have life without the software instructions.
Questions out here in the audience? Raise your hands. I can repeat questions back so that we capture them in audio. Just raise your hand and I will find you. How are we doing, Joey?
We're doing pretty good.
All right, not pretty good from Joey Ito, ladies and gentlemen. All right, let's talk about the MoMA. Can you imagine some blue tagged DNA from Venter's team or anybody else in the MoMA collection?
As far as I'm concerned, yeah, absolutely. But I'm not the only one making decisions. And, you know, ideas about design are, you know, $1 a pound. I have my own, and I've been trying to push it, and it includes the blue cell because it's blue. I mean, if it were just like a cell, there, you know, I don't mean to be facetious here, but design has...
Hugh would be pushing it, is what you're saying.
Design has some formal intention. I really do believe that intention in form, which doesn't mean beauty, which is a strange word anyway. Intention in beauty is a form of communication. Intention in form is a form of communication, right? So because it has this kind of evidence unto itself of being blue, like the blue cells, the blue DNA has taken over completely, then yeah, it can stay in MoMA. If it were just a little more anonymous and just, you know, less explanatory visually, it could be somewhere else. But I like that statement.
That's a new threshold for us.
Yeah.
Yes. Well, you could certainly tell the management at the MoMA that it wouldn't require an entire gallery
No,
to
I know.
display the...
But you know, we already had at MoMA objects that were alive. You know, it was not really synthetic biology, but almost. You know, we had work by Symbiotica, the lab in Australia, that had done this in vitro small coat made of mice stem cells that was growing. So we can get there. But you know, can I ask one question? Because you were talking about problem solving during your talk, which was really interesting, because problem solving is almost a cursed definition for design. I mean, really suffocating. And I try to say to people, no, sometimes design is problem making and then solving it, but it's also envisioning the problem. How did you deal? You seemed to be almost happy to have instead that definition come into you, or that kind of action being prescribed to you.
It's my definition of what science really is, actually. It's solving problems either on the scale of understanding how something works or solving problems, how to apply it. I think the definitions, the way science is taught, is really a false system. And most scientists spend their time solving problems, whether they're for the planet or for the daily problem that occurs.
So for you, it's a very pragmatic science. It's like a design kind of science.
Yes. Well, in problem solving, you have to be pretty creative most of the time to solve these major roadblocks. So.
Let's talk a little bit about your minimal biology notions here. When I was growing up in elementary school, there were these creepy pictures in encyclopedias that talked about what would the threshold be for life, and there were, you know, tide pool puddles with like lightning coming into them at some sort of Precambrian period where somehow the electricity jazzed this sort of pool of sludge, and creatures came out of it, and that this was somehow the epic event that crossed the threshold from chemistry to something more like life. What do you suspect that minimal biology will be, and what are the triggers in both the chemistry and the behavior of these molecules that you envision that might actually answer that question?
Well, let me make a very clear distinction. What we're doing is starting on top of 3.5 billion years of evolution. We're not trying to recapitulate what might have happened 3.5 billion years ago. There's some great scientists doing that. Jack Sozak's team across the way here is trying to get minimal RNA molecules that can replicate in a lipid vesicle, trying to see if that could be a recapitulation of early origins. We're absolutely building on top of the 3.5 billion years. What we're making are modern, very complex organisms that are nothing like what early biology would have been. So they may give us hints of what minimal functions are now, but even the most minimal cell with several hundred genes is a very, very complex entity with things that have to work far more complicated than most things that people deal with.
Is there a quality in physics here in the sense that in the same way that physicists understood that there were mathematical limitations and prescribed parameters associated with the standard model in nuclear physics that related to how many quarks there could be and what sorts of pairings would have to happen, that the minimal life problem as you see it would set those same sorts of parameters in biology in the same way that we understand some of the same parameters in physics?
I think that's a fair analogy. We're trying to get to, we're building a minimal cell. There's no such thing as the minimal cell, because you can substitute sugar metabolism with another type of metabolism. But there are fundamentals for self-replication of how the cells have to divide, how you maintain the membrane integrity. The most complicated system we have in all biology is the ribosome complex, that as that chromosome is being read with a messenger RNA, there's already a huge ribosome complex within milliseconds hanging off the end, making proteins out of that. That took a very long time to evolve, unless wherever it evolved, whether it came here by panspermia or evolved here, that's almost as complex as any cell on its own. And you know, these are not trivial processes. Once we have the fundamentals of those, adding variations to the theme is going to be the easy and the fun part.
Panspermia is not a men's club in Charlestown.
What the hell is
that? Could you explain what panspermia is all about a little bit?
And this is supposed to be an educated group.
Yeah.
It's life originating somewhere else and impregnating the planet.
I got you. Thank you, Joey. Nice. Very well done. Best moment of the Google Jock Day here. What scares you the most, Paola, about the kinds of technologies and approaches to design and nature that you've heard or seen here today?
Not much. No, that I've heard and hear, no, nothing. What scares me is the lack of critical tools that are taught. You know, education scares me. You know, there's really, there's no education about design whatsoever that is given to children, and there's very little education about science given in the right way. So that scares me, because once you have education, you know what to push back and what to accept, and you know how to metabolize innovation. So that's what scares me, not what's going on here.
Craig,
I couldn't agree more with that.
Listening to Neri's presentation, where you have a very kind of macro view of what's beautiful, how can we understand what's beautiful about the process that we see in nature, how can we respect that process and simulate that sort of thing. When you hear her talk and her approach that's very sort of stand back at some of the processes and objects in nature that are at a much bigger scale than you look at in molecules, what lessons, what sort of cross-disciplinary potential do you see there with an idea like that, or a scientist who looks at nature in that way, or even the way that Hugh looks at science?
Well, I saw it as a wonderful appreciation for the complex structures that do occur in nature. But I very much like the 3D robot, and I was trying to think, if we put our synthetic cells in that and print those out, can we make biological structures that are living buildings in a totally different way than a skin that changes color with time or something. So my imagination was running wild during her talk.
Architects are going in your same direction. I mean, architects and designers. So that's why design, architecture, and science, besides art, art's been a partner for a long time, but are really coming together.
All right, what cell would you like to synthesize based on the kinds of functionality you see in nature? Is it a nerve cell that's been talked about today? Is it a nutrition cell, skin cell, blood cell? You talked about blood cells being incredibly complicated.
So I think the problems we're facing in society right now, I have an equation that I usually put up: food equals water equals energy. And we passed the 7 billion people mark last October. In 11 years, we're going to add another billion people to the population. 12 years after that, we're going to add another billion people. So we're talking about adding the equivalent of China every little more than a decade to the population. So
a lot of DNA.
If we can, it's a lot of DNA, but it's sort of repetitive DNA. What we need is DNA that changes everything we do and how we generate food, how we generate water, how we generate fuel. So if we can design, which we've made some excellent progress on it, a cell that can modify photosynthesis, get all its energy from sunlight, all its carbon from recycling CO2 instead of trying to bury it in the ground, and make food by eliminating cows and chickens and pigs, eliminating farming as we know it, which were great for early primitive societies, don't work too well when you have 10 billion people on the planet. Those are the kinds of cells we need if we're going to survive long term.
Paola, you want to give us a final thought here? What is it that inspires you most about nature? Since I asked everyone that question, you don't have to answer that, just a final thought.
That inspires me the most about nature. I'm inspired by how people react to nature. You know, it's both positively and negatively. I've been thinking of an exhibition called Broken Nature. I don't know when it's going to happen, which is not anymore about organic design imitating nature in this lyrical, idyllic way, but rather trying to fix what was broken by wrong mistakes, but mistakes of the past. But whenever I see the work, both the bombastic and glorious work of some and instead the minute and archival work of others, I realize that nature is just the ultimate resource, and I am happy that we didn't make it so we can continue being inspired, because it's so much bigger than us.
And it's really an ancient narrative that sort of defines what we've discussed this day. If Aristotle's prescription that all art must be imitative of nature is something of an homage to the complexity of nature by individuals who could barely understand it in the way that we understand it today, science comes along and we have more of a reductive, structural trying to understand what's going on with nature. And then we finish with a sense of, no, actually, maybe we can intervene here and improve nature, collaborate with nature in some way. What an extraordinary day. I thank, first of all, Joey Ito at the Media Lab and all of the fine people that helped to prepare this event, especially my colleague and friend, Hugh, Herr. Thank you all for an extraordinary day. To both of you, thanks. And there's more activities, a reception. You're welcome to explore the lab, but Inside Out is out for now. Thanks so much.